So thank you, everybody. Greetings to all from Nairobi, Kenya. Welcome to the sixth seminar in the series Crop to Hunger Case Studies in Africa and Beyond, supporting CGIAR partners through genotyping services. Thank you very much for your time and for your presence. So some of you stay in the previous seminar, and we had Saleh Sood, that presented us with interesting updates on breeding and genetics of potatoes. And it was really a great presentation. So today we have today with us Dr. Patrick Ogum from the Cowpea program at IITA, who will be presented by Dr. Tim Close. So for ones that do not know Dr. Tim Close, which is difficult, he is a distinguished scientist and geneticist holding the title of Emeritus Professor at the University of California, Riverside. He has extensive expertise in the field of cowpea and has dedicated a significant portion of his career to collaborate with us in Africa and with the other parts of the world. So Dr. Tim, thank you very, very much for accepting this challenge, and the floor is yours.
Okay, thanks Anna for that introduction and for this opportunity to introduce Dr. Patrick Ogum, who works with Dr. Usman Bukar at the Institute of Tropical Agriculture in Kano, Nigeria. For my part, I just want to give some background on my own involvement with cowpea, most specifically development with Bukar and Patrick of the relatively new mid-density SNP genotyping capability that is now available through the Excellence in Breeding platform. Then I'll turn it over to Patrick, who will provide additional insights on cowpea and the use of genotyping more broadly. First, I'll make a few comments on cowpea to put this into context. Cowpea is in the species Vigna unguiculata, which has several subspecies, including subspecies unguiculata, which is the cultivated, usually grain-type cowpea. Cowpea is in the family Fabaceae, along with many other legumes. It is in the subfamily Papilionoideae and the tribe Phaseoliae, which encompasses several other warm season legumes, including, as shown here, common bean, soybean, and pigeon pea. And there are numerous other warm season Vigna species, such as mung bean, adzuki bean, bambara groundnut, moth bean, and numerous others. There are some good reasons why cowpea has received attention as a crop plant in the USA. It's not so widely known among contemporary plant scientists that cowpea was among the very first plants to be studied genetically way back in the early 1900s by Spillman and others to understand the inheritance of seed coat color and patterns. But even before then, way before, in fact, it was known in the USA as a reliable crop plant for seed and forage. As noted here, the name cowpea seems to have come from a message from Vice President Thomas Jefferson, who later became President of the United States, to a former senator from Virginia, John Taylor, where he wrote, "I have also received all the good kinds of field pea from England, but I count a great deal more on our southern cowpea." Both men were landowners with farms, and cowpea was farmed in present-day USA since at least the late 1600s, after being brought in probably with the slave trade and by Spanish colonists of the present day into the present day southwestern USA. In the USA, we're most familiar now with cowpeas as dried grains with a black area near the hilum, thus the name black-eyed peas, which go by other names such as southern pea or crowder pea, and in Africa there are many more known. Cowpea was grown on more USA acreage than any other legume until the 1930s, when soybean became more prominent. And there were about 5 million acres of cowpea in the USA at its peak. That has declined to less than 100,000 acres in recent decades, though there seems to be a resurgence of interest in this inherently environmentally tolerant plant, attractive not only for its grains, but also as a ground cover and forage crop. But really a big reason for the funding of genetic research on cowpea is its relevance to global food and nutritional security, particularly in sub-Saharan Africa, where cowpea is a versatile food crop crop plant in sub-Saharan Africa. It's a major source of dietary protein for people, providing lysine and tryptophan to balance the amino acid composition when paired with cereal plants. It's also a good source of folic acid, which is important for proper neural tube development in newborns. I have several images here, one from a grain market in Nigeria, one lady selling fresh pods along the roadside in Senegal, and another akara, which is made from a paste of pulverized cowpea, sold along a roadside in Nigeria. As Patrick will describe in more detail, in Africa, cowpea is consumed not only as a dry grain, but also as a fresh leaf, or as a young green pod, or a green piece shucked from the developing pod. And the leftover parts of the plant can be used as animal fodder. The cowpea is also important in cropping systems to help maintain soil fertility. The current day uses of cowpea in sub-Saharan Africa have driven the design of the mid-density SNP panel for the Excellence in Breeding platform by virtue of long-standing partnerships between researchers at UC Riverside and West African breeding teams that date back about 45 years, including my own involvement since about 25 years ago. And I'll summarize the elements of the mid-density platform now. So the SNP genotyping panel that was developed for the Excellence in Breeding platform ended up carrying a little over 2,600 SNP markers. And given the size of the cowpea genetic linkage map in centimorgans and its physical size, this is an average density of about 3 SNPs per centimorgan. I note there's someone waiting. Okay. And about 4 SNPs per megabase pair throughout the 11 cowpea chromosomes. These SNPs were chosen as a subset of approximately 50,000 SNPs that are on the Illumina iSelect cowpea consortium array that was developed in 2014 and described in a 2017 publication from our cowpea team of researchers at UC Riverside and breeding teams at IITA in Nigeria, INERA in Burkina Faso, ISRA in Senegal, and CGIAR SARI in Ghana. So selection of the SNPs for the mid-density platform began with SNP data generated using the iSelect array from 2,714 diverse cultivated cowpea sessions. In the prioritization for the mid-density platform, extra weight was given to 184 accessions that are commonly used in the breeding programs in these same 4 West African breeding operations. Some additional filtering criteria also were applied to SNP selections, with a few exceptions just to fill gaps. Missing data rate in the iSelect less than 5% was one criterion. Minor allele frequency at least 0.2, which was true for all but 21 SNPs in the mid-density platform. And even spacing along the genetic linkage map by centimorgan, weighted slightly by local recombination rate, such that lower density SNPs were planted in low recombination regions. This also involves some back and forth and testing with Andre Killian, who runs DART in Canberra, Australia. The final design was a total of 2,753 SNPs in a DART tag test set against 376 cowpea DNA samples provided from IITA. The result, as noted above, was 2,602 good SNP assays in the mid-density panel, which Patrick made use of in various analyses that are included in his presentation that will follow. So to wrap up my introduction, I want to note some funding sources because this new resource represents fulfillment of promises that were made at the outset of several projects to generate real outputs of funded research, in particular, in this case, to generate, to enable adoption of genotyping as a routine tool in West Africa and elsewhere. The three initiatives were the fountainhead of this trek into development of the 50,000 SNP Illumina isolate Cowpea consortium array, which then enabled the derivation that I have described of the approximately 2,600 SNP mid-density panel for the Excellence in Breeding platform. And these are the Innovation Lab for Climate Resilient Cowpea, funded by USAID from 2013 to 2019. The 2014 Illumina Greater Good Award to reduce the cost of the isolate consortium array, and a project through the NSF BREAD program, advancing the cowpea genome for food security from 2016 into 2021. And now at this point, I turn you over to Patrick Ongom with my comment that I'm very pleased to see that these efforts to make SNP genotyping broadly available and generally useful in the hands of talented colleagues are coming to fruition. And I stop there. Thank you.
Thank you, Dr. Tim. Patrick, are you able to share your screen and presentation, please? Thank you. Kindly put it in, yes. Okay, thank you. You are muted.
Right. Excellent introduction by Dr. Tim. Really privileged to have this opportunity to share what we're doing with the Cowpea low and mid density marker panels. Thank you everyone for joining us today, and I will take you through how we have been deploying these marker panels. First, I will give a brief background, and Dr. Tim has already shed a lot of light on Cowpea, so this will be very quick and highlight some objectives. I will give some quick outlines of the genomic resources for Cowpea and how we are incorporating this into the Cowpea breeding program. And then I will dive into what we have done with the low density marker panels and the mid density panels, and we will conclude from there. This has already been highlighted, a very key food security crop, Cowpea, and self-pollinated diploid species with about 640 million base pairs, and with the origin traced back to Africa. This key crop, which is a major food security crop in Africa and elsewhere in the world. Production covers about 14 million hectares globally, and the area planted with Cowpea, and annually production is about 8.9 million metric tons globally. In sub-Saharan Africa, we have about 7.8 million tons of Cowpea that is produced annually. In the farmers' field, however, we still have low productivity, coming to around 600 kilograms per hectare. So Cowpea being very significant crop because of its versatile habits growing in marginal soil, and also very inherent tolerance to heat and drought, is cultivated in more than 80 countries worldwide. And Nigeria so far is the largest producer and consumer of Cowpea. It accounts for about 46% of world production. So it's a key crop, and there has been a lot of attention to this crop because of its nature in fitting to the climate scenarios that are coming these days. In terms of breeding, the crop is challenged by a number of constraints, ranging from parasitic weeds like striga. We have a lot of viruses that attack this crop, and diseases, bacterial blight, and several others, and insects is devastating on this crop. Maroca is one of those, aphids, thrips, and even with its level of relative tolerance to heat and drought, extreme heat still cause a lot of yield losses in cowpea. And because of this, the breeding program has been focusing on developing short, medium, and late duration cowpeas that have enhanced resistance to some of these constraints, striga insects and diseases, and also focusing on attributes of high yield potential with adaptations to the hot and the dry zones in the tropics, and the market preference that have been targeted the medium to large wide-seeded cowpea varieties, mainly for food and feed. And this is where the breeding program is focused on. And given all these challenges, there has been a lot of efforts to try to develop genomic resources that can accelerate the breeding process. So over years, there's been a lot of advances in developing genomic resources for cowpea, which Dr. Tim and the colleagues at UCR cannot detach from this process. They've contributed immensely to building the foundational genomic resources that the breeding programs are exploiting at the moment. We now have a reference genome for cowpea that has been developed based on the IIT variety 499.35, and recently additional 6 genotypes of cowpea have been assembled and annotated, and this has constituted a pan-genome of cowpea, which actually provide a good representation of the genetic attributes of cultivated cowpea. And in line with effort as well, there has been development of genotyping platforms, and the one that Dr. Timmons mentioned, one of those is the iSelect array, which has a little bit about 51,000 SNPs that has been exploited a lot in the cowpea breeding program for mapping traits and other application. There's also another set, the Golden Gate array that is available that has been exploited with about 1,000 SNP. And we also have genotyping platforms like the DATSEC from the Australian team, which is a GBS equivalent that the cowpea groups are exploiting also for genotyping. And recently, the mid-density genotyping platform has also been developed with over 2,000 SNPs, and we'll discuss a little bit more on top of what Dr. Tim has already presented. We also have the low-density marker panels, which are basically for quality control and trait-based markers that has been developed, and we will dive into them also in the coming slides. How are we trying to combine these genomic resources or incorporate these genomic resources into the breeding program? This is just the strategy that we are doing. So the cowpea breeding program as a stage get process that we follow from stage 0, which starts with market intelligence and looking at product profiles and looking at key traits that are demanded by the farmers, and the traits that require discovery are identified from the product profile, and it goes into stage 2 where there is a lot of this discovery. This is where a lot of molecular applications is placed, looking at pre-discovery part, where we are looking at assessing the genetic variations in the germplasms that are available, which also provides sources of donor parents that are used in the breeding program. And the data accumulated from those characterizations put into a trait discovery process through running a number of duos or linkage mapping to identify interesting alleles that are needed in the breeding program. And those go through validation, and eventually validated markers are deployed in the breeding program. Stage 3 is the line development process, and sometimes the low density marker panels are also used at that stage to carry forward some of the lines that are rich in the alleles for the traits that are of interest in the breeding program. And stage 4 is when we are starting early testing, and as you'll see in the discussion later on, is where we want to initiate the genomic selection. So that's where the mid-density panel will be highly useful. And so the marker systems are integrated in the program, starting from the high-density markers which are needed for discovery, and then we have the low-density panels that are converted into breeder user-friendly, mainly for quality control and for selection purposely. So let's look at the low-density marker panels that we have. There are two types, as I've mentioned, the sets for quality control and then trait-specific markers. These are all based on the competitive allele-specific PCR. And briefly, without going into details, the CASP assay has normally 2 forward primers, which targets the biallele, the 2 alleles in the SNPs, and then 1 common reverse primers. And each of these primers are labeled with the fluorescent dye that allows excellent detection of the genotype. And then it goes through the normal PCR process for amplifications, and it's easy to detect the genotype calls with the fluorescent flaws that are at the end of these primers. So if the 2 homozygous allele, blue and red, and when it's heterozygous, it overlaps and it produces a green flaw, and so it's easy to call the SNPs and call the genotypes. And so that's how the process that the QC markers are following. And so the QC panel that we are working with now is about 17 SNP panels that was originally developed by genotyping about 177 diverse copy lines with the Illumina Golden Gate. So you can see we started mining the resources that were developed earlier by our partners. And so, and then it went through selection processes, looking at polymorphism, SNP neutrality, heterozygosity, and then genome coverage. And so this process allowed us to identify a set of initially 19 SNPs that could be used for quality control in the breeding program. Those 19 went through SNP verification process, CAS assay verification process. And in that process, we are looking at those allele SNPs that can differentiate clearly between the homozygotes and heterozygotes. And through that process, we are able to drop 2 of the markers, and the 17 SNP markers remain that were very effective in differentiating between the homozygotes and heterozygotes and making them more of good quality for use in the breeding program. And so recently we tested the effectiveness of this panel in the large scale application in the breeding program. We're trying to test them for hybridity authentication. And so we made about 225 unique cross combinations. We had about 220 parents that were crossed to 2 sister lines, and that gave us about 1,000 F1s that we use this set to test how effective this 17 QC panel would work on a routine basis. And we got some interesting results. So initially we are checking the efficiency of those QC markers, and by efficiency here we are referring to ability of those SNP markers to differentiate between the parents of a cross. And so on the graph on the left there, you can see mark efficiency on the y-axis, and these are the 17 SNP markers that we tested. And generally, most of the markers were able to differentiate very well between the parents of a cross. Is basically the polymorphism between the 2 parents, and there were just a few that perform less below 10% efficiency. We also looked at how these sets of QC panel could help us detect relatedness between parental lines. And, the focus is always not to avoid crossing sister lines. So we wanted to check if this QC panel can help us differentiate between parental lines that we are putting in the hybridization program. And purposely, we included those some sister lines so that we could know if it's doing a great job. There were also some accession, some land races that we included. And we found that the panel was able to classify the population pretty well and put the sister lines in the same cluster. And then these were the rest of the breeding lines that were recycled for hybridization. And so we found that the QC panel was working pretty well. Further, we checked if the QC markers could also be useful in detecting the impurities that are in some of the lines. So we compared the F1s and their parents on your right here. So looking at the proportion of heterozygosity on the y-axis here. And then we have the F1s and the parents. And as expected, you can see that the QC panel were able to detect that the F1s are highly heterozygous. It's what we expected. And the parents were mostly homozygous, but it was also still able to detect some residual heterozygosity in some of the parents or issues of admixtures in the parents. And so it's still telling us that the QC panel can help us make very good decisions in the breeding program and make good progress. And we also checked out of those 1,000 F1s, we ran the QC markers on them, and we wanted to see how well it could identify true hybrids and versus the self-progenies. And then you can see that for each of the crosses that we had there, we had to scan through, just check if the marker is differentiating between the two parents. And in the example here, you have this parent cross to a local land race, and these are the SNPs, the SNPs call. Okay, so, and then these are the progenies. And so if you scan through, you can find that the F1s for each of the locus are heterozygous until when you get this progeny number 4, which is homozygous for this SNP locus. It's also homozygous for this SNP locus. It's also homozygous for this locus. And therefore, you can conclude that this is a self-progeny and the rest are actually true hybrids. And so with that, when you have a large data set, you have to derive some statistics that can help you quickly scan through the large germplasm. So we computed the degree of hybridity of each of the F1 and use it to sort the progenies quickly. And our assessment, we found that the panel was able to detect true hybrids, but 79% of the F1 that we had were true hybrids, and 15% were selves that we could detect. And then we had issues of missing data, where you could not confidently determine the hybridity of a particular F1 because you have a lot of missing data across that particular F1. And then there were a few cases where the markers could not differentiate between the two parents, and therefore you also don't have high confidence to declare that these are true hybrids or not, because you don't have polymorphism between the parents. But basically, generally, it was quite useful. The panel is very useful in identifying hybrids. Recently, we've deployed this in also a bigger panel with our NAS partners from Ghana, Burkina Faso, and Nigeria, and trying to support them through the Crop to End Hunger project, support them in making decisions on progenies that they can advance in their breeding program. And so you can see this is just a snapshot from the Ghana partners that we try to evaluate their F1 progenies based on the level of hybridity. We are able to recommend the crosses that could go forward. We did a similar thing to check the purity of the lines. So they had some breeding lines that they were not sure, and they wanted to know if there are some admixtures in them. And so the QC panel did a great job, because you can see, like in this first set here, we take 10 samples from the same genotype, and then we genotype them with the QC panel. And if you see in the first set of lines up here, these first 10, they came from a single genotype, but we treated them as independent samples. We genotyped them with the QC panel, and you can see that we are able to detect that they were 100% homozygous, but we also looked at the similarity among them. We ran the similarity index, look at genetic distance between them. We are able to discern that, okay, all these 10 are genetically similar using those QC panel. But when you come to the second set down here, as an example, you can see we also have 10 samples here. The level of homozygosity started varying, right? Some are 100% homozygous, some are not. And then they may be 100% homozygous, but check if they are genetically similar. You need to run that genetic similarity analysis. And then we can start seeing that the similarity index is not zero. The genetic distance between them is not zero. So that already signals that there is some kind of admixture in this line. And so it calls for purification. So you can see that the QC panel are able to give us a light on what to do and the decision that we need to make in such scenarios. Actually, if you scan also through the sequences, it's normally difficult when you have a lot of germplasm to go one by one on the sequences, but the summary statistics can help a lot to quickly make the decision. You can go through each of the SNP that we have. We would expect that because these are the same samples, we should see the same allele, like GGG throughout, but you can see from here, we run GGG and we start seeing AAA. That means there is a kind of genetic differences here. So this set of this line is having some admixtures at this locus, so at several locus, and I just gave a snapshot of the genotype data. And so the QC panel was very useful in identifying breeding lines that are mixed, and then we could make a decision on how to purify them, and that would help the breeding program a lot. Still on the low density marker panels, we started by assembling traits that have been mapped. Some we did in-house GWAS mapping, and then there are those that were previously mapped and from literature. We assembled a lot of SNP markers, and all this mapping were based on the initial genomic resources, either the iSelect or the Golden Gate assay that we are used to map this, and we have information on the SNP markers that are used and even their design sequences. So we assemble all these trait-specific markers, and we came up to about 75 trait SNP markers, and Anna was really very instrumental in supporting us through this process. These SNP markers were converted to CASP assay, and to do that, we to verify that those markers would work properly in the population. So we use about 17 diverse Cowpea lines, and we included technical replicates. Each of them were replicated so that we can be sure of any errors, any genotyping errors or DNA quality issues and that. And then we also purposely included some F1 progenies to be sure that the markers would be able to distinguish between homozygotes and heterozygotes. And these were sent to Intertech, and CAS assay were developed. So the verification report, we found that most of those 75 were good, except a few. So you can see the good ones are able to clearly distinguish between the homozygotes. The red and the blue are the two homozygous SNP alleles, and then the green are the heterozygote. Then we had a few that they are bad. You can see that it cannot differentiate, and so those would not be useful markers. Some were inconclusive, and so out of 75, there are just 3 that fail the process. And so we started the process of validating those markers. So we created different populations with different genetic backgrounds from the populations that were used for mapping. So we've created about 10 different populations that we are advancing, and currently we have the F4, and at F5 or F6 we will be genotyping all these populations with the 75 markers, which are targeting different traits. Striga, aphid, thrips, seed size, seed tolerance, and we will phenotype populations as well, because we want to check the consistency of these markers in tracking these traits before we can deploy them. And so we're moving with that process almost towards the end. There were two sets of traits whose markers were previously developed that we have taken a little bit far, the aphids and the bacterial markers that were already also converted to Cas markers, and we created some unique populations to try to validate this, but also we are doing two things. We are kind of deploying it in a forward breeding approach, and we also want to check if the markers are still tracking the traits pretty well. So we have populations that were developed purposely for bacterial blight, and then we have additional populations for aphid for aphids. And initially at F2, we genotype them at F2, and then advance those plants to F4, and at F4 we re-genotype them. So we are carrying forward both the plants that carry the alleles for those two traits, but at the same time we also take forward some of the lines that don't carry the alleles, because we wanted to validate, to check if the markers are really tracking the traits properly. And we've initiated the phenotyping also of these populations. The aphid, we have an efficient aphid phenotypic platform, and we are phenotyping the 1,000 lines that are coming from the aphid populations. And then we are also phenotyping for bacterial blight. And this will just be used for confirmation that the markers are correlating with the phenotype. The lines that are coming from those are going to be handing over for advanced yield trial, or some of them that are really consistent will be taken used as parents in the breeding program to improve these two traits. So that's how we've gone with the low density marker panel. The mid density marker panel, thanks to Dr. Tim, is already given an elaborate explanation on how it was designed, and about those 2,600 dart tag SNPs that were derived from the iSelect array. So what we did, we tried to test the performance of this mid-density panel under different scenarios. And the first thing that we did was to check if it can be used for population structure studies and the genetic diversity studies. And so we in the set that was genotyped, we included different sets of materials that we knew a little bit about their genetic background. So we had some accessions, the gene bar accessions, and some of them are the one we commonly use in the breeding program. And then we also purposely included some biparental rails, about 100 of them in that set. And then there were a set of breeding lines that we also included. And then there were a group of magic lines that we had that were also included. These sets of materials, we basically know their background, and it was good that we put them together and we see how the mid-density panel is going to classify them. And interestingly, when we run the cluster analysis, and you can see we tried both PCA and DAPC, and you can see how the panel is able to classify this population. The biparental lines stood separately, clearly distant from the rest of the populations, and this because one of the parents of this biparental was a wild relative, and you can see that it really separated itself from the rest of the group. And then we have the multiparental lines. These are the magic that also separated itself from the rest of the group. The distance is not much because some of the magic lines are also part of have been part of the breeding lines that were used to develop those magic. And then we have the breeding lines and the accessions. A lot of accessions here are normally used in our breeding program, and you can see that they became closely related, but there was a clear distinction classification that was telling us that the panel is very suitable for genetic studies and structure analysis. We also check if the panel could could assess accurately linkage disequilibrium decay and trait mapping. And again, from what we already know from the genetics of this population, the biparental reals have less recombinations, and therefore the rate of decay would be low. And this was detected clearly by this panel. See, the blue is a slow decay in the biparental reals, and then the accretion and the breeding lines that have a little bit of time to for recombinations, it was decaying faster, and this was expected. We tested, even though it is a medium density marker panel, we tested the possibility of still using it to hypothesize genomic regions that might be associated with key traits in breeding programs. And it was interesting that we had data on seed and flower color on this population, and it was able to remap the flower and seed color position that was previously mapped in Cowpea. And that gene is responsible for pigmentation in Cowpea. So it was interesting to see that the panel is working effectively and can also actually be used in trait mapping. And we also try to check if some sets of these can be used for quality control. We use the entire set in a way, but it helped us actually to identify a few sets that we could use to enhance our quality control marker panel. So on the left here, we are looking at a test for polymorphism. We are looking at on the x-axis the percentage of marker polymorphism between the parents of a cross. These are parental combinations. These are the F1s and their parents that we included in the genotyping panel, so that we will be able to see if the markers are working well. And so we could see that the panel is able actually to differentiate between the parents of a cross more effectively, with polymorphism detection ranging from 16 to 61%. That means that the panels, out of the 2,000 markers that you have, those are the percentage that we are able to differentiate between the parents of NA2 cross. On your right hand side, we have the hybridity test again using the entire set, and then we also, since we included the F1s in there, we're able to assess the level of hybridity of the F1s using that panel, and it was able to really discern the differences. Those that are true hybrid, you could see the level of hybridity is going up to 97% hybridity of the F1s were able to be assessed. And so with the computation of hybridity, you can set a threshold and say only those with hybridity above 70% are the one you have confidence in declaring them as true hybrid, and then there were those that we could declare as false hybrid. So again, you can see that the mid-density panel can be used for a diversity of applications in the breeding program. And finally, we are beginning to initiate genomic selection process for Cowpea, and as all of us know, for genomic selection, you need a prediction set that is both genotype and phenotype, and then that helps to generate the marker effects that is later used to predict the selection population. So for Cowpea, this is what we are proposing. So that the genomic selection can fit in our normal breeding program, because we normally start by making crosses, and that goes from the parental generation, generating the F1s, and goes through the process generation advance. We derive the populations from F2, keeping about 300 to 500 F2s, and those are taken forward through single seed descent until F5 or F6. That's when we start our yield testing. And we want to initiate genomic selection at this point, so that if we can predict lines with high genomic estimated breeding values, those would be recycled back into the hybridizations. And then the normal selection continues also to lines to advanced yield trial. And eventually those can also go in for variety release, national performance trials and release. So what we have done so far, we had about 800 breeding lines that were at PYT. So we genotype all those 840 breeding lines using the mid-density panel. And what we have done is just playing with that marker data. We have used that marker data to try to get a subset that we can phenotype more accurately to give us to become our training set with a sufficient phenotypic information. And we would use that to develop the prediction model that will routinely be used before the materials reach PYT. We would genotype all of them and use our model that is accurately built based on sufficient phenotypic data on the training population and the genotypic data. And that would allow us to predict materials that we can use as parents. And if possible, some of them could go further for advanced yield testing, and also can constitute candidate varieties for release. So we have identified a set of 200 lines that we have initiated the phenotyping process. We want sufficient phenotypic information because this would be our key prediction set. And with the preliminary data that we had on the 800, we thought of checking just the accuracy of prediction. There was just 1 year data set. And so we just ran the GBLAP model and did some cross validations with K folds of 5 and 20 iterations. So with K equals to 5, that means the entire set was subdivided into 5. And then a lot of sampling is done. You make one set a prediction, and then the other set is a selection set, and then try to estimate the breeding values and then run some kind of correlations. And so for this preliminary exercise, we saw that so in this radar plot, the traits that are outside this cycle are the one that has high prediction accuracy. Grain yield had slightly lower prediction accuracy based on this preliminary data. And then we saw that maturity traits, this to 50% maturity was better. It had better prediction accuracy together with 100 seed weight. And so this just giving us an idea of how the mid-density panel is going to function in a genomic prediction approach. Otherwise, we are still phenotyping our training set. And I think the prediction accuracy will be much more improved with a better phenotype on the training set. And so that's how far we've gone with using the mid-density set. There are a lot of other applications that are being used. There are populations that we've created to try to map traits using the mid-density sets, and those are in the pipeline as well. So then in summary, we can see that we have made significant progress, in developing, validating, and deploying the low and the mid-density marker panels for Cowpea. The markers are now used for parental selection. That's using parents that are not genetically related. We are using these resources for routine F1 verification, both in our program and in our partners program, the national programs, line purification, genetic diversity studies, trait mapping, marker-assisted selection, and this is going to get even better when we validate a number of trait-specific markers that we have developed. And then we just started to initiate genomic selection. I think this is the end of my presentation. I would like to acknowledge our partnership with Tim and Dr. Tim and the colleagues from UC Riverside, the funding, from Bill and Melinda Gates Foundations. A lot of donors support the breeding program, and we would not want to end without acknowledging them, and support from the Excellence in Breeding platform. And Anna has been very close to us in this process of developing the markers, the marker panels. And so we really appreciate all the supports and contributions to the Cowpea breeding program. I want to end here and thank all of you for your time and attention, and I would ask if there are questions. Thank you.
Thank you, Dr. Tim. Thank you, Patrick, for the excellent presentation. So now we can go to the questions, and I will ask to Bodo to unmute himself and to ask the questions that he wrote in the chat. Bodo?
Yeah, hello. Thank you very much for this nice insight you have been giving to us. Great. It's just great. I think this, yeah, you have made good progress, and this is really a lot of useful tools, and it's quite nice to hear about this. So now my specific question was when you Patrick were talking about your selected subset of markers for QA/QC, and then you extended to other sets with more markers. I understand these are CASP, so they are probably the most affordable technology, which is very good. I'm still interested in knowing what is the cost of this, what cost is caused to the program, what subsets or sets of materials at which stage in the breeding are being tested as a routine. And so what benefits did you get out of using these marker sets? And relative to the costs, is this something that is economical? I hope this is clear. Over to you, Patrick, I guess.
Good. Thank you, Bodo, for this question. Yes. So we have a subset of QC markers. There are 17 at the moment. They were carefully selected, and so they do a great job. It wouldn't hurt to increase the number a little bit, just in case there are situations where you have diverse populations that were not included in that test set, and therefore you will start seeing some monomorphic kind of SNP calls coming in, and so it's fine to increase that number from 17, and actually in the set that we use for the NAS program, we actually added some trait-specific markers into that QC, and it did a great job.
Patrick,
yes.
Patrick, excuse me.
Yes.
My question was not about whether this is, needs to be increased. My question would tend to be more toward the direction, is 17 markers necessary, or could this number even be reduced? Because, from what I've seen with other crops, 17 is already a considerable number of markers for this routine procedure that has to be included everywhere.
I get you. Yes. Yeah. So I was coming there. So 17 for us, I think, is good enough. Actually, I wanted to increase it because you've seen from the performance of those 17, in some cases, it fails. There are issues. There are issues of monomorphic, and then there are issues of missing data that are coming either from quality of DNA or from the technical issues in the SNP calling. And so if you have very few, then eventually you will end up with a situation where you cannot make decision because almost all of them have not worked. Either there's missing data, then you end up with one or two markers only that are showing polymorphism between the parents, then you're not confident, right? So that's why at least, 17 is good enough. If I can go to 20, it's even good enough. The cost for genotyping is not that, is not very high now. I think Anna can speak a little bit on that, but I think it's a combination of the number of SNPs and then the number of samples that you have, but it's not beyond $3 a sample for the low quality for the low density marker panel, and so the cost is within range, right? And so I think the marker panel should not be extremely small, because you end up with some issues of making decisions, and if you have missing information. I don't know if I answered you fully.
Yeah, thank you. Yeah, I still think $3 per sample is very expensive from an economical standpoint when you are in a company. Okay. You have to count every cent, in fact. And that is why, you know, it's a feeling from my side, because I don't have any evidence. That is why I was relying on you. Maybe you can look back at the tests that you've done so far and somehow come up with, you know, also when you talk to the breeders or when the breeders talk to you, what is necessary? Because some of these things can be sorted out once you know the direction. For example, when you know how much contamination occurs, then you can design your testing and the numbers in a way that you capture most of it. Okay, there will be maybe a little loss, but then it will be much more economical, and it is clear. This is just the thought behind that. Thank you.
Yep, I agree. Thank you. Yeah.
Thank you, Patrick. Petra and Pernilla from Intertech, they are here. Maybe Petra, if you don't mind, do you want to comment on the pricing of the CASP SNPs?
Yeah. Okay, thank you, Anna. To Bodo, we at Intertech won these CASP at a very cost-effective price, actually, and something what I think is so nice with this project, and what I hope that Patrick can also, when he does all his calculations later in stage, is that when you have these trade markers for traits which are very expensive to test in the field, it actually, you know, when you make the whole sum of how much it costs to do all these field trials and how much time it does, compared to running these CASP assay and these panels for these traits, it's actually much more cheaper and faster to run these CASP assays. About quality control, it really depends of how important quality control is, and how fast you want to be, and how good control you want to have to check if these CASP assays are expensive or not. Right? I think that as an answer.
Thank you, Petra. So we also have a question from Christian. Christian, do you want to unmute yourself and ask the question to Patrick, please?
Sure. Well, it's not a question, it's just a quick comment. First of all, Patrick and team, congrats on the development of the SNP array, which seems to work pretty well in identifying the right parents and the right germplasm. I just wanted to point out, a lot of you probably have seen the pyramid that we use at Accelerate to show the individual steps of improvement, and you might remember that genomic selection is at the very top, and that a lot of other things need to be in place to make sure genomic selection is working well. Running genomic selection is easy. Making sure genomic selection is a cost-efficient tool that gives you more gain at the same or even a lower price is really challenging. And I don't want to beat around the bush here. From what I've seen in your presentation, there are a couple of things that would make me be concerned about this being the case. So long story short, reach out to us at Accelerate, and we make sure together that you get the most of your strategy of what you want to achieve. There were a couple of things, again, like the training population size, like how often you want to update, etc., that may not really give you what you want. So again, just reach out to us. It's not a discussion topic for today, and we make sure together that it's gonna run as efficiently as possible.
Thank you very much, Kristen. We'll do the needful.
Thank you. So before we finalize this presentation, Petra, you put something in the chat, but it's very small. I can't read. Do you want to make the question, and after we go for the terminus of this seminar?
Oh, sorry if it looks so small. No, but I really like this collaboration. Thank you very much, both Patrick and Tim, for a very nice presentation and showing how you use these SNP panels. And what I wonder, I think it's so interesting to see that Tim is working with the Southern American cowpea, and you, Patrick, are working with the African cowpea. And I wonder how much can you learn from each other? How much can you use these panels together? Or are they very different for your material? Or is it a 1 + 1 is 3, right? So how do you work together, and how can you learn and make each other better? That's what I wonder.
Right. Tim will probably add, but what I want to say is that the panel was developed based on a diversity of materials. Some weight was put on African germplasm, but there was a diverse collection of materials that comes globally. And so the panel seems to be very effective globally, actually. So it's a very useful panel because it has a broad background from which it was developed. I think Tim can add more explanation to this, if it's still around.
So yes, I agree with what Patrick said. So to answer the question, UC Riverside began to be involved with West African breeding programs in the 1970s, and has continuous engagement, especially with groups in Senegal, Nigeria, Ghana, and Burkina Faso. Previously, there were also some involvement with researchers in Angola briefly, and Cameroon, when Bukar was in Cameroon before he went to IATA to lead the program there. There's been numerous joint publications on mapping traits and establishing seed purity, and developing new varieties that have been released in different countries. Some decades ago, there were varieties released in Senegal. It was a partnership through my predecessor, Tony Hall, who began the breeding program in Riverside in the 70s, and former leader of the Senegal program, Ndiaga Cissé, where the new varieties were released in an area where the villages that adopted the new varieties had seed production during a time of drought. They were basically early varieties. And for that, Ndiaga Cissé was given a national award from the president of Senegal at the time. So these are just some examples. There have been, I think, well over 100 publications that are jointly authored by researchers at UC Riverside and West African breeding teams over the years. So there's many traits where genetic mapping has been done, and so now that means there's a repository of knowledge of regions of the genome, and therefore markers, nearby markers that can be used and integrated in the breeding programs. So it's kind of there'd be a long story to try to be comprehensive in an explanation, but I guess the idea would be it's in the literature documenting these interactions, and so much of the research, much of the funding from U.S. Agency for International Development came through Michigan State University. A mandate was always to provide relevance directly to the USA, in addition to being relevant to development of cowpea agriculture in West Africa. So I think that's probably about the best I can do to answer that question, Petra. Thanks.
Thank you, Dr. Tim. Thank you, Patrick, for the excellent presentation. Thank you for accepting this invitation. So before we finalize the session, once again, I want to remind all of us the reason of these seminars. So these seminars serve to highlight our colleagues' work, the good work that everybody is doing, through also our support, but the support also of other donors. So we want to acknowledge this support, that without it, it would not be possible to be here. And we can't also forget the rest of the team of the breeding resources. This include Eng and Rajaguru that are also supporting these activities. And also the vendors that are also doing a lot of effort to this be possible. So we came to the end of the 6th seminar of this series. We thank you all for being with us and hope to see you in our next seminar, where we will have Gloria Adu from the CSR Ghana to update us on her recent research in maize in West Africa. And she will be introduced by Dr. Cláudia Guimarães from Embrapa, Brazil. So thank you very much to all and see you next time. Thank you, Dr. Tim. Thank you, Patrick.
Thank you. Thank you, everyone.
Thank you.
Thanks.